what we are witnessing today is a culmination of our process that started close to 4 years ago. And I'm happy that today, as we are gathered here today, we are witnessing an event that declares that that process was actually successful and it was implemented.
This project is global engagement and research and experience for undergraduate students. And I am the PI of the project. It is funded by National Science Foundation. The overall goal of this project is to expose our students to a global setting and also research. It is becoming very evident that competitive students are going to be those with a more broad global outlook on research and research policies. And so we wanted our students to do research, but in a global setting, hence the host, which is Egerton University, for which we are very, very, very grateful.
During the 2 months, the students had the opportunity to be attached to mentor professors, where each student had an opportunity to carry out individual research. We had an opportunity to expose these particular students to some of the top-notch lab facilities in Kenya. They also had an opportunity to visit research stations other than Egerton University across Kenya. And finally, they had the opportunity to engage with professors at Egerton University for specialized professional development activities.
When I first began this project, I really didn't know what I was expecting, because I am a biomedical science major and I am mostly focused on cardiology, and I was not really seeing myself working with agriculture and food security, but here I am at the end of it all.
Actually, I didn't see myself doing stuff like this. Ever since I started doing this research, it's actually been an eye-opening to me. It's been like teaching me how actually food safety is something that everybody needs to pay attention to. And I know this is something I would take back home to my country. I'm from Sierra Leone.
My experience in the lab has been very formative, like working odd hours to make sure the project is done, and being passionate enough to finish this project with such great goals.
So my project is focused towards the livestock industry in Kenya. It is a growing industry here that is very important to many small stake farm holders all over the country. So prussic acid is a form of cyanide that when it enters into the animal system can lead to asphyxiation and in extremely high levels death. So I've been able to evaluate from sorghum plants at the 3rd leaf stage grown here on Egerton University's campus, and found that at this certain stage that they are not at lethal levels, but that eventually the local varieties that most farmers in the areas grow have had the elevated prussic acid levels that can lead to damage to the livestock. And then also looking at lignin and cellulose content. Lignin and cellulose are structural components that have an inverse relationship. When lignin enters the animal system, it cannot be digested, but cellulose can be digested in livestock that have the rumen capability. The results have been very good, as cellulose content has often outweighed, especially again in the local varieties.
And we are looking at how we can improve sorghum varieties to fit in with our country's need for feed industry. And the student that was attached to me, Mr. Luke, has been working with me for the last 5 weeks. And I've enjoyed working with him. He has been a very good student. What I like about him is that he has a very good attitude to work. And we have worked together within the shortest time he has been here, be able to generate some data on this research?
We drove to one farm in Rongai and another farm in Naishi, which is very close in Njoro, Kenya. We sampled the lab and we took the nodules and we isolated and characterized the isolates onto Congo red, Burmotamo blue, heat labile, and heat stable carbohydrates. And we also did with antibiotics. And with all of that, it took the entire experiment, but we actually got to a result in which we were able to characterize 2 superior strains of rhizobia from the Rongai region that was able to inoculate under every single plate possible that we put it under.
And so we were able to select these dolichos from Central Rift Valley, and Kiki was able to isolate rhizobia from the dolichos and characterize it using morphological and biochemical approaches. And this is a project that I've always wanted to do within my lab, but for one reason or another, we had not been able to do it. But with Kiki being in the lab, we were finally able to do it.
And for further research, what I would hope is that they would actually plot these specific accessions of rhizobia onto fields to see how they grew and then place them under environmental stresses.
We've been looking at how different environmental stressors can affect growth and nodulation of the lab crop, which is an important crop, but it's being underutilized here in Kenya. So farmers are cultivating this crop less and less because of economic constraints and the fact that it's very labor intensive to grow. So far, we found that a lot of the variation in how the lab grows under these environmental stresses actually has to do with the variety. Once we know what environmental stressors each variety can handle, we can better match a farmer's land to the kind of lab that he or she can grow. So that way we can improve yields and overall food security for Kenya.
So Kiki was able to isolate based on different carbon sources, the utilization of rhizobia of different carbon sources, and also the ability of rhizobia to grow under different concentrations of antibiotics. Both characteristics are very important in being able to identify a superior strain that can be used, hopefully in inoculants to boost the production of dolichos.
The aim for my project is basically to look at the determination of anti-nutritive in spider plants. And what we're trying to do is to help food security and food safety in Kenya, because when you impact a lot of spider plant, it can be harmful for your body.
So we are working with Cleom gynandra, which is popularly known as spider plant in Kenya, which is a popular vegetable, especially for lactating mothers because it's very high in calcium and has a very good complement of vitamins in it. We were looking at anti-nutrient factors because these are popular vegetables, originally weeds, but they have now been upgraded to vegetables, and we need to know if there are any anti-nutrients that might be available or in high quantities in them. And so we were working with these students to look at that. This has been work that is continuing. We've collected many ecotypes from different regions of the country, and we are characterizing them, first morphologically and then genetically, and these students are now looking at biochemistry.
We want to be able to utilize a vegetable named spider plant to help fight malnutrition in Kenya. But it's also important to know if there's any anti-nutritive factors that might inhibit the full utilization of the plant. So we ran tests on tannins and alkaloids to see tannins are an anti-nutrient that inhibit protein digestibility, and alkaloids are anti-nutrients because they are known to disrupt the nervous system and also inhibit and cause damage to DNA. It is important to know the levels these anti-nutrients are found in the spider plant, and then the spider plant goes in various environments throughout Kenya. So we want to see if there's any correlation between environmental factors and the levels of the anti-nutrients.
It was a good experience working with these students. Short time, but very intense work was done, and we were able to come out with results. Overall, we wanted to analyze up to 37 actually, ecotypes that we had, but we managed to, Egerton managed to look at 19 and Egerton at 21. And so work is going on these
on these vegetables, and we will be happy to host more students in the program.
I conclude the best protein source was snapping green early in compared with the other crops in the Central Rift Valley of Kenya.
Finding the best ratio for complementary foods, I found that KNE 741 was the best in the low altitude and that P224 was the best in the medium altitude, and would like to continue my study by doing a sensory study which will allow the mothers of the children to try the complementary food, which is like porridge, so that we could further analyze the benefits and the quality of the food that was created.
We've been working on finding a way to combat the fall armyworm problem in Kenya. The fall armyworm is a pest that invades a country. It's invaded Kenya, and the larva destroys crops, particularly the maize crop is what we're concerned with. The maize crop is basically, it's a cash crop in Kenya. It's a huge economic factor. So finding a way to combat the fall armyworm is going to be beneficial, because that means improved yields and it's going to improve the economy of the country. We did several tests. First test was to find out which pheromone lure was the best for a trap. The second one was to find out if the traps had any effects on the surrounding plants within 10 meters. And from our findings, we found that we did find the lure that was the best. They attracted the most moths. But we found that the pheromone lures, the presence didn't really affect the surrounding plants. Like, they were infected at the same rates, even with the lures around.
I've been focusing on the research of the fall armyworm because it has a lot to do with food security in Kenya, and that is the focus of our work here. The crop in particular that we're interested in saving is maize. It's critical to consumption for Kenyans. We're looking to define what is the genetic strain of the fall armyworm that is present in the fields of Kenya. If we can determine what the strain is, then we're able to contribute that towards science, so that engineers, agriculturalists, and those who are developing pheromone and lure traps are able to better optimize their methods by which they create those devices. So far, we have performed some DNA barcoding on a few fall armyworm samples that we found throughout many fields, and we're awaiting with great anticipation to find the results of what genetic strain is here.
Besides doing the research, we've trained them also on communication techniques, and for which reason every Tuesdays they meet for research forum where they present their progress, and also professional development sessions every other 2 weeks. So we have been training them all to be scientists in all aspects. Today is the concluding day, and we're just so grateful. They have prepared their posters. Everybody is here to talk to them, and I'm so proud of the students. The students actually come from 6 universities. I have 9 students from 6 universities in the US, 4 from Bowie State, being the host university, and the others are from Indiana University, Oklahoma State University, Farmingdale in New York, and I have one person from Morgan State University, and also all the way from Puerto Rico. So we are just so blessed. These students have worked so hard. And in conclusion, I also want to talk about their cultural experience. We gave them a time to work and interact in a different culture. We took them to different places so that they can experience different cultural activities, including Masai Mara Game National Reserve. We took them to the hot springs in Lake Bogoria, I believe. They had a wonderful boat ride in Baringo and many other places. They also visited some other exceptionally good labs in Kenya, including the ILRI, International Livestock Research Center, and ISIPE, International Insect Research Center. So it has been a wonderful experience. We hope to come back next year bringing another 8 or so students. And I'm just so grateful to Professor Gendo for all that he has done, grateful to the mentors for all the great work, grateful to ARC, the hotel that has hosted us. Thank you to the transport section. They have gone with us. The drivers have been patient. Everybody has been excellent. I hope I didn't forget somebody, but if I did, please know that you have been wonderful. Thank you so much. We look forward to next year and even a greater time. Thank you.
The one thing that I loved about this experience was that I got to work with Godelieve and Shadrach and Professor Nancy. When I tell you that they dropped everything, anything that they were doing, to come and help, to come and make sure that everything ran smoothly, and I appreciate that to like the grandest amount.
I really enjoyed getting to work with Shadrach, Professor Nancy, and Godelieve on this project. They've been very helpful throughout the entire duration of this project.
And working with Professor Richard and Milka and Cyprian has been really, really great. I'm really grateful for them. Thank you for staying in the lab with us really late at night. Thank you for putting up with us, being patient with us. Even when we call you and text you, you're always there for us. I just want to say thank you to Egerton University for opening their labs, their gate, and being a great host to us.
It's been very fulfilling working with Professor Richard and like learning how to perform our research and making sure like I'm on time, I have the resources that I need, and performing the research to the best of my ability.
I want to say thank you to the National Science Foundation for funding this project and provide a research experience for all undergraduate students. I want to say thank you to my mentors, Professor Kimurto, Cyprian, Tracy, Dr. Osano, and I'm so grateful for being here. Thank you so much.
We're very fortunate that they've been so welcoming and hospitable towards us and accommodating. We're thankful for the wonderful collaboration that we've been able to experience and witness this summer through them.
And I just want to say thank you to the National Science Foundation for the funding for this grant to make it possible, to the friends that I've made here in the country of Kenya, and my American team that's come with me. Asante sana.